Hello everyone, welcome to my channel. Let's talk about botulism today. So, botulism is mainly caused by Clostridium botulinum, which is a gram-positive and spore-forming obligate anaerobic bacilli. It produces a powerful toxin called botulinum toxin, which causes botulism, a rare disease manifested as various clinical syndromes ranging from food poisoning to wound infection to infant botulism. The term is derived from a Latin word called botulus, meaning the sausage, a poorly cooked sausage formerly associated with the food poisoning. So mainly we are going to discuss 5 main things under botulism, which are pathogenesis, clinical manifestations, types of botulism, laboratory diagnosis, and treatment. It is ubiquitous in nature, widely distributed as saprophyte in soil, animal manure, vegetables, and sea mud. Let's talk about its pathogenesis. So, Clostridium botulinum is non-invasive. Its pathogenesis is due to the production of powerful neurotoxin, that is botulinum toxin, probably the most toxic substance known to be lethal to mankind. Serotypes are, there are 8 serotypes: A, B, C1, C2, D, E, F, and G. Out of these 8 serotypes, A, B, and E cause human disease, most severely being serotype A. All serotypes produce neurotoxins except C2, which produce enterotoxin. Botulinum toxin types C and D are bacteriophage coded. BT differs from other enterotoxins that is produced intracellularly. The botulinum toxin is produced intracellularly and not secreted and appears outside only after autolysis of bacterial cell. Toxin is synthesized initially as non-toxic prototoxin, and it requires proteolytic enzymes to convert it into active form. So the mechanism of action of botulinum toxin is it enters the blood circulation and then to the peripheral cholinergic nerve terminals, and then it inhibits the ACH receptors, which eventually produces flaccid paralysis, and it does not affect the CNS. The therapeutic uses of these flaccid paralysis can be the treatment of spasmodic conditions like strabismus, blepharospasm, and myoclonus. And this toxin is also produced by Clostridium butyricum, Clostridium barytii, Clostridium argentinensis. That's about the pathogenesis. The clinical manifestations are especially due to the reduced ACH acetylcholine in cranial nerve and parasympathetic nerve. So, manifestations appear after the incubation period of 18 to 36 hours. Diplopia, dysphagia, dysarthria are the manifestations. You can remember them as D cube. And then descending symmetric flaccid paralysis of voluntary muscles, deep tendon reflexes, constipation, no sensory or cognitive deficits, and respiratory muscle paralysis, which eventually leads to death. These are the clinical manifestations. And then let's talk about the types. There are mainly 6 types. Number 1 is foodborne botulism. It results from consumption of foods contaminated with preformed botulinum toxin. Most common source is homemade, improperly canned or fermented food and beverages. And then most cases are sporadic. Outbreaks are very rare. And wound botulism It results from contamination of wounds with Clostridium botulinum spores. It presents like foodborne botulism, and it can be recurrent in injection drug users, example black tar heroin. And then infant botulism, which causes floppy child syndrome. And adult intestinal botulism, iatrogenic botulism, and inhalational botulism are another types. So let's go to the laboratory diagnosis. The laboratory diagnosis mainly involves isolation of the bacilli and toxin demonstration by mouse bioassay. Isolation of the bacilli. Usually the serotyping is done by type-specific antisera. The culture is especially by anaerobic culture like RCM, Robertson-Cook-Mead broth, and this Robertson-Cook-Mead broth culture, growth on culture media, can be confirmed by gram staining and biochemical tests by automated methods such as Marlitov. Automated tests like Marlitov and gram staining, it is gram positive and non-capsulated subterminal or oval bulging spores. Then toxin demonstration is by mouse bioassay method. The toxin is inoculated into mouse and the animal is observed for paralysis. This method is more meaningful. And then let's go to the treatment part. The treatment of respiratory paralysis is intensive care support such as mechanical ventilation. Next, botulism antitoxin. It should be administered immediately on clinical suspicion without waiting for the lab confirmation. The antitoxin neutralizes the unbound free toxin molecules. Next, in wound botulism, abscesses are clean, debrided, drained promptly. Antibiotics such as penicillin and metronidazole may be useful in wound botulism. So that ends the complete explanation. I hope you liked it. So please like, share and subscribe to my channel. Thank you.